A very good morning, everyone. I, Adhya Sethia, welcome you all to the final day, day 4 of IONS Manipal 2019, with the hope that we were successful to make this journey of yours a memorable one. It will be our utmost pleasure and priority to end this day with a bunch full of memories. Without further delays, I begin the day with a scientific talk of Dr. Kevin Ellisery. I request Ms. Wenzel to come here for his introduction.
A very good morning to you all. I feel utmost privileged to introduce the first speaker of the day, Dr. Kevin Ellisery. Dr. Kevin Ellisery received his undergraduate and graduate training in microbiology and biotechnology at University of Wisconsin in Madison. He worked in Professor John White's laboratory, developing imaging approaches for model nematode C. elegans. He received his further postgraduate training at National Integrated Microscopy Resource in the area of computer science and microscopy. He is currently director of the Laboratory for Optical and Computational Instrumentation and a principal investigator in the Laboratory of Molecular Biology at the University of Wisconsin in Madison Graduate School. He also directs a collaborative medical engineering group at the Mogridge Institute for Research. He has over 100 publications ranging from informatics and instrumentation to live cell microscopy and cancer imaging papers. His current research focuses on the development of novel imaging methods for investigating cell signaling and cancer progression and the development of software for multidimensional image analysis. Today, he is here to share his knowledge on imaging fingerprints in the cell world. So please join me in welcoming Dr. Kevin Ellisery to the stage. Over to you, sir.
Can you hear me okay? All right, good. I talk very, very fast, so if I get too fast in the back row, I can go like that and I'll slow down. The more enthusiastic I get, the faster I talk. Well, thanks everybody for staying for the last day. I'm delighted to be here. It's my first time in India, and had a lot of Indian postdocs and students. They're all excited I'm here, and I'm equally excited to be here as well. So thanks for having me and inviting me. I really love giving talks with students. I'm around all day if you have any questions about the research or working in the US or imaging research in the US. I'd love to talk about that. I'm originally from St. Louis, Missouri, and I came to Wisconsin in '92 expecting to stay for 4 years, and over 25 later, I'm still there, and I love what I do. I got sucked into the world of optics and imaging. I started off as more of a cell biologist, but then I got optical and computer training, and very excited about what can be done in cancer and other diseases with these technologies. My talk is really about the idea of looking at the cellular world, of trying to understand what happens at the cell level, particularly from the perspective of human health. We often think about tissues and organ scale work, but everything good or bad in your life happens at the cell scale. Death and birth, these are all cellular events, and understanding them is really fundamental to human disease, both basic research and even clinical. So we're talking about some new work in this area that we're trying to do to understand this idea of cells, and particularly the idea of microenvironment. Microenvironment has often been ignored. Over the last 25 years, we've often taken cells in the natural context, put them on flat pieces of glass, and image them. But the microenvironment is the intact world they live in. You're my microenvironment. The room lights, if you guys fall asleep, you impact me. If the room lights, if it gets too hot in here, you impact me. This microenvironment impacts my behavior. The same thing with cells. If you want to understand cells in the natural context, you need a realistic microenvironment. And that's the other piece of my talk. So for some time, my lab has tried to understand this microenvironment in the context of live specimens and live cells. And the one common thread of our recent work, which I think many of you also work on, is wound healing. So trying to understand what happens with a wound. And again, a wound is a central part of almost every bad thing that can happen to you. A wound, of course, can be a cut, can be an abrasion, but also can be cancer invading. It can be diabetes. Wound can be a burn. Wound can be things that perturb your normal state. So trying to understand how cells in the microenvironment work together to actually combat or go after wound healing effects is a big thing of what we do. And a big part of what we do, I'm not going to talk about today, except for the last slide, is software. So about half the lab develops software. for two main reasons. One is interest, but the other is fundamentally that is where the field has gone. When I first started my lab, software was a small part of the work. You needed software to actually analyze the data. And back then we even had some microscopes that weren't even digital. So you had to convert analog to digital. Now everything is digital. But with that comes an opportunity that to analyze the data as it comes across the microscope, not just after the fact, but while you acquire. So this idea of computational optics, that the software could play a role even as you acquire to get better data sets. So better software, probably the best known example is ImageJ and Fiji, which we developed, and I'll give one slide at the end about that. And then a big part we do is image informatics and hardware to do all these various things. Spatial temporal, which I'll talk about, looking at space and time, looking at spectral lifetime, which a couple of professors here work on, the idea that you can measure intrinsic chemical events. So for the first time with an optical microscope, you can do more than just look at GFP or rhodamine or your favorite extrinsic fluorophore and see what happens with your eye or with magnification, but you can see the chemical reaction. We can measure for the first time things like pH and hydrophobic and hydrophilic and other kinds of reactions with a microscope, which you normally would expect that only a biochemist could do. And then large 3D and XY montages. This is the idea that if you went to like a major monument and you wanted to see everything with your iPhone or your favorite camera, you can stitch and you can get a panoramic view. We want to do the same thing, except we're looking at a tumor. If we want to get the whole view of a tumor, the idea of doing a really large stitching, or in our case, it could be thousands of images stitched together, and I'll show a few examples of that. And then one thing I won't have time to talk about is multi-directional. All the work that I do, and I think a few other talks you saw, we're looking at the sample en face, up and down through me. But you don't get to see the back of my head, you don't get to see the true 3D nature. So this idea of light sheet microscopy or optical tomography, where you spin the sample, and that's something else we're starting to work on. And then multi-scale, I'll talk briefly about. This is the idea that you want to link the worlds of imaging. So in my department of medical physics, we actually sit by spatial scale. You'll see one table of ultrasound guys, one table of MRI, one table of PET, one table of optical, which is like me, one other guy. And it shouldn't be that way, right? The way that cancer works, it doesn't respect these multi-scale fragmentations. Cancer happens across scale. It goes from micron, submicron and up, and we want to go after that. And I'll give some examples. And so this is what we're trying to do in our group. Today I'm going to talk about the blue part, which is what I spent most of my career on, multiphoton, confocal microscopy, this next talk, electron microscopy, AFM. And the reason microscopy techniques are so popular is that ability to get submicron resolution. And in many cases, be able to get live imaging at that. So you can look at things at a nanometer scale. So with the multiphoton imaging we'll talk about, you can see changes at 300 nanometer in intact samples over time. But we can't actually look at a whole human. We can't see a whole heart in a human. Our ability to optically section and resolve large tissues is limited. So that's where anatomical imaging techniques like MRI and CT and ultrasound are so powerful and are used in pregnancy and cancer because they have a resolution and depth that we can't get in optics. And then metabolic imaging is where you can really see powerful changes in glioblastoma in the brain, for example, be able to look at metabolic changes in whole brain, whole tissue. Again, something that we can't do on that spatial scale. But it's important to note that if you look at this slide, I got double mics here, you see that I drew it kind of funny. Maybe you first think that's just a Photoshop typo, but there's this really interesting thing in our field that I always want to get across, that not only is this whole field developing, and we're always working on new improved methods, but there's a massive mesoscale gap that no one in the field has solved. We actually are pretty blind from about 10 to 100 microns. There's actually no good ways to image that. There's promising techniques like photoacoustic imaging, for example, but really still very much out there what to do. And so that's something I really get across to you, is that not only can you innovate in a spatial scale, but there's actually a whole unknown spatial range that people have not looked at with modern imaging. And so for our work, I spent most of my career in the microscopy realm, as have several other speakers, because of the ability to get this great resolution, but at a great price. So that resolution I want to see, because I want to see cell-based changes. And these cell-based changes, you can't see with ultrasound OCT. They're too small, they're sub-micron in many cases. But the price I pay is sectioning depth. And so as you look at the work I'm going to show you, we're limited. So it means that we use animal models like mice, rats with a window we implant. I'll talk briefly about that. We're limited to zebrafish and C. elegans, samples that are clear, or we're limited to samples from humans that are biopsies. But we can't put a whole human on our microscope because that's sectioning depth issue. And that's why there's a real need for developing other methods and trying to register them across these spatial scales. Let's give some examples of what you can do in cancer. drug delivery with OCT, which is a type of optical method in between ultrasound and us, and of course, medical imaging. And so to sum up kind of the optical introduction, because I'm not sure what you've heard, so I want to make sure that you kind of know where things are at. Optical microscopy has come a long way. Just in the 25 years that I've been involved, it's really, really exciting how far the optical microscope has come. It still is very much the tried and true benefit of actually being able to look either at a grayscale, non-label sample, a fluorescent sample, so genetically tagged, looking at time lapse and structure. So many times people use optical imaging to just watch a mutant over time and see what happens. So watch a C. elegans mutant, watch the cells divide, or watch a tumor metastasize. So the unrivaled ability of optical imaging to look at time and space is very, very powerful. And the major reason people do it, particularly with the advent of fluorescence. So the reason confocal microscopy, which is the next speaker, is so powerful, it's its ability to get high resolution images of cells over time and space. But these other two categories, chemistry and physics, you may have an idea what I mean. You may be thinking that he has chemistry there because of the chemistry of fluorescence. So to put a GFP in a sample, you have to understand the green fluorescent protein, how it works, what its excitation and emission are, the fluorescent properties. And the physics would be the physics of the microscope. There are reasons that microscopes look the way they do. There's a reason a lens is the design it is. There's a reason there's a certain length to a microscope. And if you ever wondered all microscopes look alike, there's a reason for it. There's actually physics of that design. But in this case, these two yellow tabs here, chemistry and physics, I have up there to kind of set up the rest of my talk, because these aren't meant to be about the actual microscope or imaging modality. I'm actually referring to the signal. So the same way that like a GFP in a worm or a fish watching cell division or a cancer cell metastasizing would have intrinsic dynamics and structure, I want to get across to you today that there are many examples that they also have an intrinsic chemical signal and intrinsic physics. So I'm suggesting to you that you can use a microscope to measure physics, to measure chemistry. And how can you do that? Well, quick examples are you can look at stiffness. You can use optics, tell apart a tumor that's more stiff and not stiff. And it's known that stiffness plays a role in breast density for women. Density of the breast is an important factor. Then in chemistry, by measuring special properties that I'll describe called lifetime of fluorescence, we can tell you which sample is more or less pH. We can tell you something's hydrophilic or hydrophobic. If you're a biologist, we can tell apart a lipid raft from a non-lipid raft. And so this idea of being able to look at intrinsic properties of your cellular environment without any labels is something that gets us quite excited, because it makes it clinically relevant. So intrinsic properties is what we're going to talk about. And then, of course, in this kind of talk, we have to gloss over the details. People often really stress the hardware need to do that. These fancy multi-photon microscopes that I'll mention, laser scanning microscopy, the fluorescence lifetime hardware, photon counting optics. But the software is really a big, big part of all this. So the major part of my talk I decided to pick is the idea of the tumor microenvironment. And I very briefly set it up that you're my microenvironment, and this microenvironment is different than the tumor microenvironment, because hopefully you guys are not hostile at me. We're a friendly group, and you guys are hopefully happy I'm here, I'm happy I'm here. And so it's a good microenvironment. This is the normal microenvironment. But with a tumor, this microenvironment is one where it is trying to hijack your body. It is an adversarial microenvironment, and it is one where the tumor is trying to get an edge. It's trying to take advantage of your natural body's microenvironment. It's trying to take advantage of what's in your body to hijack and try to spread in your body. And the cancer's ultimate goal is to spread and metastasize in your body. And this is just a small subset of all the things that make up the microenvironment. There is a very famous set of entity called the extracellular matrix, which has, unfortunately, until about maybe 15 years ago, really wasn't widely studied. And then adipocytes, fibroblasts, immune cells and tumor cells, and then, of course, the macrophages and collagen I'll talk a lot about. And if you look at the papers going back the last 50 years, the thing that always gets studied are the tumor cells, which really doesn't make any sense. If you just want to understand a tumor cell by itself, tumor cells will act differently if they're by themselves versus if they're in a natural microenvironment. And the example I often give in my classes is if you were asked to write a book about how Kevin, or a paper about how Kevin spends his week, if you were approached like a cell biologist over the last 20 years, you would hire a team of investigators to take me out of my house in the middle of the night. You'd bring me to a warehouse, it could be in India, a warehouse somewhere the non-disclosed side of town. And that warehouse, I'd be in a room and there'd be a one-way mirror. And you'd put me in that room with nothing but bread and water. And for the course of the week, you would film me behind the window what I'm doing. And I would spend the entire week crying, right? And I only do that one day a week, right? And the only one. And that sounds silly, right? But that's what we do with cancer. We rip it out of its natural environment, we put it flat on glass, and we image it. It's wrong on so many levels. One, cancer cells are never alone. What about normal cells? What about the idea of having different ways of imaging it? What about trying to actually give it more natural nutrients? So to make that environment more realistic, if you went back to the example with me, what if you allowed me to have a TV, a sofa? What if you allowed me to interact with some friends occasionally? What if I was given more stimulating food? The chances are that in vitro environment you created wouldn't be as same in my normal life, because I wouldn't be walking around seeing all the different buildings and friends I always do, but you'd see a subset of it. It'd be more realistic. And that's our dream, is to have a more realistic microenvironment. And that's what all these guys are. These are its friends. These are the things it needs in this particular case, and also the things that it's trying to combat against. And in this case, you want the tumor cells to lose. But I really do want to stress is that you have a realistic microenvironment. And that's why the field is starting to move away from flat on glass, 2D cells flat, and are looking at the most realistic microenvironment. If possible, live in the human. But if not, what about human cells in a collagen gel? What about human cells in microfluidics, those of you that buy technology? What if you can create engineered models that inspire from the human? We can even 3D print tissue now. What if you can 3D print tissue and put cells back in that? Or what if you can image live in an animal? What if you could put human cells in a live mouse or a live fish, which we can do now? So this idea of a realistic microenvironment is very important. So I think you've all heard about fluorescence. Fluorescence is what I work on. For most of my career, I've worked on fluorescence, and I think everybody knows here, it's a major, major breakthrough. People discovered that this green fluorescent protein in jellyfish could be engineered and put into human cells, engineered models, zebrafish. This idea that a gene of interest could be tagged, not only with that animal habit, but all its progeny. So this idea of being able to do follow things genetically with labels is important. But my twist on it is that I want to study this, and I want to use that really value of that extrinsic fluorescence, so the fluorescence you add to a human model, using GFP or using some other favorite engineered protein. But I also want to take advantage of the intrinsic state. And there's two intrinsic things I want to harness. One is I'll talk about how I want to actually take advantage of this excitation to a higher state of fluorescence and measuring how long the emission lasts. And I'll describe that process we call the lifetime of fluorescence, why that's important. But I also want to do something that I want to actually measure the intrinsic fluorescence. So the same way that GFP was native to jellyfish, and we saw, the original scientists, Marty Chalfie and other colleagues, saw the jellyfish glowing in the tank and glowing green. We want to look at, in our body, proteins that glow. And if you do have any questions during this conference, there's this amazing site. Unfortunately, he passed away last year, but a good friend had this website called Microscopy that was 10,000 pages. This is Michael Davison at Florida State. Over 10,000 pages of microscopy information. So if there's anything at this conference you've heard, chances are if you Google what we're talking about, it'll be in there. And it actually will probably be better than what I presented. So I highly encourage you to look at that. If you recognize any of the animations I have, that came from there. So it's a great site. So these are an example of some of the things that naturally glow in your body. So when you think about imaging with fluorescence, you normally think as a very foreign process to human imaging. But in humans, not only can you tag cells with GFP or some other protein of interest, the exciting thing is there's lots of things that glow in your body. And as a cell biologist, we were taught to hate that. It was really annoying when you're going to image your sample. There's all this background noise. So if you had GFP imaging it and you excite GFP in cells, all these other things would glow, right? But it turns out that if you understand what they are, you start getting excited. You start recognizing the names as a biologist. You're like, wow, NADH and FAD glow, collagen glows, elastin grows, lipofuscin grows. There's a growing list of over 25 proteins that naturally glow in the body. Why is it exciting? Well, from a translational point of view, it means that you're allowed to do things clinically you couldn't normally do. For the most part, fluorescence is not allowed clinically. But if it's already there, you can use it. And also it means that if you are studying a process, nature's been very kind to naturally label things so you can follow their contrast. So the rest of my talk is really be about how we can exploit that endogenous contrast in the cellular microenvironment to understand a process like cancer invasion and progression, or also known as metastasis. So a good part of my talk is about multiphoton. I'm not sure if you've heard about that yet, but a real quick intro, I think maybe a little bit of a glimpse into that, is multiphoton is really a good analog to confocal. The confocal lecture you're getting next really transformed the way we did imaging, because confocal uses a pinhole to reject out of focus light. If you were trying to take a picture of your family, a crude analogy is if I was shining a flashlight at your family from behind and you couldn't quite see the picture well, you could use an aperture, a physical aperture, a big pinhole to reject my flashlight and see a good image of your family. And that analogy is why confocal is so powerful. We can use a pinhole to reject out of focus light and get really good images. And they use lasers to scan a raster, a square, get this image. Multiphoton uses the same type of scanning technology. Very similar technology when you go to the grocery store and you see the laser, swipe a barcode. It's the same idea. But multiphoton does one step further, is using lasers such as what Topica makes. You can use nonlinear lasers and you can actually excite a sample to a higher state using 2 photons instead of 1 photon. So almost all the imaging you're normally exposed to, like fluorescence in a wide field microscope or confocal, is 1 photon. So 1 photon is excited to a higher state. We do 2 photons. The advantage of this 2 photon excitation process is that we can actually get deeper in samples because double the photons is double the wavelength. And you could test this yourself if you had milk and water and olive oil and you had a red and laser green pointer. The more turbid sample like the milk, the red laser pointer would penetrate deeper than the green. So by having these longer wavelengths, we can get deeper. And so that's a big part of what our lab does. And then we also can take advantage of these double wavelengths. It turns out they're so long wavelength that from a biology point of view, they're past a lot of the known UV checkpoints in your body. So you don't have a lot of the photo damage you see with confocal and other things. Then also we're talking, I think I have a slide on this, about second harmonic generation, an intrinsic property of collagen. It turns out the image collagen has this beautiful barrel-like non-symmetric structure. Light hits it, you get this instant signal. And then of course, we also do work with spectral, the idea of having basically a fancy mirror, set of mirrors that can break up spectra of light. And then looking at that with this measuring of lifetime at briefly measure, where you can measure how long things last. And so our goal is to combine all these things and image non-destructively, so without damaging or killing the sample and without adding any external fluorescence. So as I mentioned, 2 photon is where you can image deep in samples with not higher resolution, but better signal to noise than other methods. And this gives an example. If you first see this in a cuvette of dye, this is the 2 photon or multiphoton sample. It's often called 2 photon or multiphoton because usually you get 2 photons, but with the right laser amount of energy, you could get 3 happening. So that's why we often call it multiphoton. And then this is the classic single photon, like you might see a confocal laser. And when someone first sees this, they're like, why is Kevin excited by that little dot? Don't I want that big band of fluorescence? But that little dot represents the focal plane where the most energy happens. And so it was so neat is I can get this without actually needing any pinhole, without needing any rejecting out of focus light. So I get a very crisp signal only where I'm focused at, which is really helpful. And so you get optical sectioning as part of this. And because of these long wavelengths, we can get quite deep in samples. Keep in mind this is optics, right? So it's deep for us, but it's still only oftentimes just a millimeter or less, right? So it's all relative. And then, as I mentioned, less photo damage. So you can get better viability because there are checkpoints in your body that are related to DNA damage. That's one of the reasons why UV light is so harmful that we want to avoid these. And these wavelengths we're talking about, instead of being 450 or 488, like often you do with a laser at the 1 photon level, we're at 900 or above. So we're past all those known UV checkpoints of damage. And then we can also generate this property of second harmonic, which again is an intrinsic property of light interacting with the collagen. I have a brief slide on that. And then I've mentioned this fluorescence lifetime thing a few times, and all I said to you so far was it's a measurement of how long the fluorescence lasts, and it's an intrinsic property of that period. And by measuring that, it can tell you what the pH or the hydrophobic or hydrophilic properties. But to go more in depth, what lifetime is measuring for every pixel how long that fluorescence lasts. And you can get a number. And so this image looks like a normal image, but it actually is an image that has basically an Excel spreadsheet behind it where there's a coordinate telling you how long the lifetime lasts. And these coordinates can be mapped, and using statistical approaches, you can map for goodness of fit. So you can use equations to really get a good curve fit. And then we even measure the background. And the background is basically what signal you get if you don't have a real sample on. And there's a lot of air, like cabling, problems in the room. In our case, the fire exit is right above the microscope. We're not allowed to move it. So we actually could pick up the fire exit sign, right? So we have to normalize for that background. It's like if you're measuring radiation and had to have background radiation. So what's already there, same idea. So this is your lifetime, but this is pretty unwieldy for the biologist to work with. So we need to map that data back to your image. And so we can basically create a color map, and then you get this map of images. And so at first glance, one could say that what FLIM tells you is it could tell you what is the difference between a GFP fluorescence and an M-cherry or some other protein. And that would be useful because sometimes proteins overlap, and another way besides spectra overlap would be good. But it'd be a limited value, all things considered. What's exciting about this value that I was already hinting at is the lifetime of a fluorophore is dependent upon its microenvironment. That was getting back to my original title where I called cellular fingerprints. What I was getting at is that in your body, there are fingerprints that change in response to disease. And so in the case of a cell, if the pH is changing, or if there's hydrophobic, or if two proteins are close together, Dr. Parasamy, who's a leader in our field at Virginia, like many of you know, works a lot on FRET. When two things get close together, they exchange energy. We can measure that by lifetime. And so this idea of a fingerprint is really powerful. But to be honest, that's not the best analogy, because my fingerprint just tells you who Kevin is. It doesn't say who I'm interacting with and who I'm talking to. This cellular fingerprint, based on these properties I'm going to talk about, can not only tell you who I am and who I'm interacting with and where I am. And that's the real payoff. Because other than biochemistry, there's no other way of doing this. And the problem with biochemistry is they can get information about the protein, but they do it in a blender. They lose all space and time reference. We can get this information about lifetime and pH and hydrophobic and binding, but we can get it in space and time. So we can tell you where and when it happened. And that's the real power. And then I've also talked about this ability to image collagen, which I'm going to show in a second, the actual images. But this is a pretty straightforward idea that imagine this non-symmetric shape of collagen. And when the light hits this beautiful structure, it bounces back at you. And this intrinsic ability, so it's not fluorescence, but this ability to use a laser to interact with this intrinsic glowing property of collagen is quite powerful, not only because of the physics of it, but because collagen is everywhere. People that are new to biology are often surprised to hear that collagen is the most abundant protein in your body. Again, almost everything good or bad in your life is collagen related. On the good side, collagen, which means Greek for glue producer, keeps your body together. If you learn anything today, that's your word of the day. Collagen means Greek, it means glue producer. It keeps your body together. Without collagen, you can't create a collagen mutant. You'd fall apart. Collagen is necessary for your membranes to form, to become a structure of your bones. It's a part of a healthy person. But it's also a leading player in disease. It's well known that diseases like osteoarthritis and osteogenesis imperfecta, any kind of bone joint disease, are collagen related. But the work I'm going to show you over the last 15 years is we've been showing with many collaborators that in about 6 cancers and about 5 other disorders, we see that collagen is a highway for the disease spreading, particularly in cancer, that it can be a way for the cells to invade your body. And that's why we're excited by this method. And again, it's exciting because it requires this non-symmetric requirement. Very few things in your body have this. Collagen is one of them. Tubulin, if you're a cell biologist, and myosin also do this, but at a much lower rate. And it allows very high resolution. So again, 600 or less resolution nanometers. And no sample processing. If I was to take a bit of your skin or your cheek cells or something easy I could access, I would see this natural property. And then very quickly, I'm going to go over this idea of multi-scale imaging that I briefly hinted at. One of our interests is not only doing multi-scale imaging, as I talked about, from cells to whole animals and humans, but we're even interested in trying to understand it in models. And so to us, multi-scale not only means looking at pieces of humans, so biopsies, but looking at the problem in vitro with engineered models and then animal models. So the example I'm going to give has all of that. I'm just going to give one example in today's talk with maybe a few auxiliary things that show the power of it beyond that. I want to talk about breast cancer. And I'm going to talk about breast cancer from the point of view where we looked at pieces of humans. We looked at human cells that we took from a human and we cultured in vitro, and then animal models. And the exciting thing is we can take those same human cells and we can put them in the animal model. So then you have the best of all worlds. So to us, that's also multi-scale. It's a different type of thing. It's multi-scale when it comes to the model and not just the imaging method. So the problem I'm going to talk about is breast cancer. And this has been work we've been doing for a long, long time with Lee Wilkie and Patty Keeley and Suzanne Ponik and Dave Beebe and many others. And this project was a long-standing collaboration that we originally started with an imager that was trying to do radiographic mammography. And we were trying to link the problem of breast density to what happens at the cell level. As you may know, for women, breast density is one of the risk factors. In many countries, and in the US and Canada, it's by law, they do this as a mandate where women are expected to be screened for density of their breasts. This happened to my aunt. She called me up very concerned because her doctor told her she had a very dense breast. But she goes, what does that mean? And the reality is we're not sure. We know that if you have a dense breast, you have a higher chance of getting cancer, but you don't have a higher chance of dying it. And so because it's not what's called a negatively prognostic factor, a factor that would kill you, it's not known exactly what that means. And so there's a lot of work to understand this. And that's where we come in, and we want to know what are the factors that contribute to this. But the point of this slide is not only to set up the breast cancer story, but also to show you the value of imaging methods. So in this conference, you've heard from many imaging people, some of us at different scales. In your whole career, you're going to run across multi-photon people, PET people, MRI people, ultrasound people. Keep in mind, no matter what they say, including me, nobody's method is better than the other. It's better for what they showed, right? So each method has its own purpose. And I think in the case of breast cancer, it's very obvious that each of these scales, radiographic tomography, MR, magnetic resonance, ultrasound, nuclear medicine like PET, light microscopy with just basic stains, molecular assays. I made a couple of molecular images yesterday. And then our work where we can use, this is the first image of collagen, where you can use collagen with your favorite probe, in this case, Syndican. All these images represent samples from a dense breast, but they're all equally valuable. They all give you a different snapshot. No one is better than the other. So not only is this an illustration of multi-scale imaging, look at the same problem, but it's also an illustration of why all the methods are needed. And the common thread of all this work, whether it's our work in 2-photon or these other methods, is that we're all hoping to find a biomarker, something that would be actionable, something that we could find to tell the doctor to do something like apply a drug. And so for most of my career, I've been focused on the first two, trying to be quantitative and accurate. So we'll talk today about how you turn an image into a number and how you can assure that what you're seeing is a real biomarker. But it's exciting in the last 3-6 months, we're starting to work on number 3. We didn't used to, but now that we have shown this change we want to talk about, we've discovered a biomarker that's the rest of today's talk. We've actually discovered there's drugs that can interfere with it. So we've actually entered our first 2 trials using that drug on human patients. So we're quite excited by that. So we now have a clinically approved biomarker to act on, which is a big new development. And so when we're talking about is breast density, as I've mentioned to you, this is a dense breast, and it's thought to be 3 to 7 times increased risk of getting breast cancer, but not dying of it. And so we've been trying to exploit this work and trying to look at the cell scale. And so in our case, we can look, and this kind of gives away, this actually kind of gives the whole talk away, so maybe it's a little early, but basically what we can do is we can look at a tumor, and we can see the collagen go around the tumor. And we can create models that are really dense collagen. So it represents a dense breast. With the ability to engineer a mouse to artificially produce too much collagen. And we already know from gene expression analysis and other types of staining techniques, we know that a dense breast largely has collagen as part of it. But what's fascinating is when we image this, the thing that we start to see that you can maybe see here, and I'll show us more than once in this talk, is this change here. Here's the tumor boundary. And you start, the cells here, kind of, the collagen here is kind of, with the cells kind of haphazard. You can see the drawing here. But then it starts to align. And what we see is the collagen go like this. And we see this consistently. The first paper 10 years ago was 300 patients. Now we've done it in thousands of patients of different types and look at women of different backgrounds, Asian, African-American, obese, not obese. And we see this efficient change. What's exciting to us is that we can measure it with software and get a realistic measure. And what's really interesting is that this measure, this property that we call collagen signature that's tumor-associated or tumor-associated collagen signature attacks is negatively prognostic. We have shown that whenever you see this, the patient dies. And we've seen it with over 98, 99% accuracy. And why is that? We have movies of cells crawling along those fibers to invade the body. So we know that it's a bad thing for metastasis. And we can do it with images like this, where we can actually get these high resolution images of the cells, a very similar idea to confocal, just with a multi-photon system. I talked about montaging. We can start doing really big fields of view. We do this in part to link to ultrasound and other methods that are higher spatial resolution or higher fields of view. But we also do it so that we can get a better idea what's happening in the tumor. And then I'll kind of gloss through this, but we use math. This is math that actually was used originally to map ocean floors. It was used to look at the topography of an ocean floor. Turns out it's very good for looking at fibers that undulate, that change. And so these discrete changes, we use curvelet math to do that. And we can very quickly divide up your image, I'll kind of gloss over this, and get an image like this, where we can start looking at all the fibers. And so now in these studies, for pathology you normally use a human. We don't for these. We now are using machine learning and automated approaches where we're looking at hundreds and hundreds of samples automatically. And the first paper we did was by humans. It was a group of 5 grad students. And they actually analyzed the data. And then we were able, fortunately, with machine learning to reproduce that at a much bigger scale. It showed not only they correct but we can get it more accurately and quicker, this technique. And we can look at this in different types of tissues. This is normal tissue. And so you can look at the change in collagen. And this is what you see with a dye. So hemotoxin eosin, or H&E, is the gold standard in clinical care. If you ever had a mole on your arm and they want to look at that, they section it, they use this stain. We can look at that and compare our methods. And we're very interested in how to make pathologists do better so we can augment what they do. And we've published papers about how we can do pathologist-driven intervention. And you can see the field of view we're doing. We're now doing 7.5 millimeters, which is quite large for optics. We can do big fields of view. We can compare. And we can see the differences in collagen. And look how striking it is, where the collagen is kind of bunchy and wavy, and it gets long and taut the more invasive it gets. So the idea of the fiber stretching. And if I had more time, I would discuss the mechanics of that. We have a lot of work now to show that this is not only real, it is something that we can engineer. So we've been able to get the cells to do the same thing in vivo. We can create engineered gels that are long and taut. We can put ferrite dust, little magnetic dust, in a gel, and by making them stretch, the cells all of a sudden invade. So there's a lot of interest in what happens, and also the signaling proteins. If you're a biologist in the audience, things like Rho and RAS have been studied, the genes behind it. So a lot of interest in what's going on here. And if I had more time, I'll just briefly go through it, but we're doing a lot of work to understand what else this does. So we've shown with our collaborator Paul Campanola, this can happen in ovarian cancer types, and see what happens between normal ovarian, high risk cancer. And then I think I might be missing a slide here. I had two other slides I think I may have taken out, but we've also done other cancers. So we've done a lot of work in pancreatic cancer, and all more recently in anal cancer, the exact same phenomenon where we see more taut fibers being negatively prognostic. And then we've also been starting to look at collagen in the brain. And you may be, why is he looking at collagen in the brain? If you know anything about the brain, it doesn't make sense. The brain doesn't have fibroblasts, which are the main producer of collagen. In the brain, usually the collagen is around blood vessels. But we were wondering is what does collagen do in the case of a wound? What's the role in the brain? So if you look at a normal brain, this shows the power of this big imaging. You can see a little bit of collagen here in green, but for the most part, you don't see it. And we were wondering if you were to wound the brain, what happens? With a hypothesis that not only do fibroblasts produce collagen, which is normal, but we think macrophages, or we know macrophages produce collagen. And so the idea from a wounding point of view, when you think about collagen in your body as role in disease, remembering more than one type of cell can produce collagen. And so what's striking to us is that we started putting electrodes in the brain. This is very commonly used to implant electrode to actually help in either measuring recordings in a mouse or restore connectivity, like from a horrific accident, maybe you lost spinal cord function. There's promise of using electrodes to restore functionality in your body, but your brain wasn't designed to be a pincushion that things put into it. And what you see are these collagen patterns up and down the electrode, striking patterns. And we've been seeing these quite consistently. We see it within 30 days. We've shown this from macrophages. More recently, we've shown it also comes from glial cells, which is another very important type of cell in your brain. And we think it maybe has to do with the hardness of the material. Actually, we have more recent data that it is. We find that if we put a soft electrode in, you don't get that same pattern. And so we think actually the way the stiffness and the hardness of the material interact with your brain may be a big part of this collagen production to the wound. In glioblastoma, so in brain cancer, we see that same thing. So it shows that the collagen story is more universal than we think, even in cases where the collagen is not already there. And a real quick aside is that everything I've talked about for the most part, collagen is a bad thing when it's been hijacked. So in all epithelial, any skin-related disease, anything epithelial, lung, anal, prostate, pancreatic, breast, more aligned collagen is a bad thing. It will allow the cancer to divide. But in the case of the brain, our work like glioblastoma, more aligned collagen is a good thing. If you see more aligned collagen, the patient will survive glioblastoma longer. Well, why is that? The difference is that in the epithelial diseases, the collagen is waiting there to be hijacked. It's there before the cancer can hijack it. It can take it over to invade it. In the case of the brain, the collagen is not being made. It's not already there. It's being made. The macrophages are making it in response to disease. So it's a healthy immune response. That's why having more aligned collagen in the brain is a good thing, because it's your body trying to fight off the cancer. So this interplay between collagen, organization, and cancer. So I'm going to shift gear in the last few minutes. Oh, I did have pancreatic cancer. I said it out of line. So this is the pancreatic cancer work where you can see the changes in collagen architecture with that. So quite striking. between normal and cancer. So the last part I'm going to talk about is our work with metabolism in collagen. And this is, again, to look at breast density. And I've already shown you some of the slides to set it up. The idea that we can classify a dense breast using ultrasound or mammography methods. And we can actually show this linked to collagen. So we have done mammographic density assays. We've done collagen extraction. We know for sure there's more collagen in a dense breast. We also know there's more fat, more adipose tissue, but definitely collagen. And we also have been able to look at mutants and collagen promoters and look at RAS and RHO. We've been able to show the link to breast cancer as well. But we've also shown this result I've already shown you, where there's this progression of 3 stages where you start seeing the collagen go around, and then you see tax 2 where it starts to go like this, and tax 3 like this. And it's tax 3 that we care the most about, this change like this. And we've been able to show that this is, this stage here is negative prognostic. We see more aligned collagen, the patient dies. And we've been able to this is a screenshot from a movie where you can see these green cells lined up along the fibers waiting to invade. And we've seen this in many cancers at this point. And then we've actually been able to do Kaplan-Meier curves to show the survival rates, how poor it is with seeing this kind of change. So now we want to move to the mouse model. So a lot of work was done in human samples, but we want to do it in the mouse model to understand better what's going on. To not only look at the collagen story as part of that natural microenvironment that I talked about, but we want to look at the other part of the microenvironment, the other part of the fingerprint that I haven't really mentioned, and that's metabolism. So metabolism is one of the central parts of your body. So besides the collagen, the rigidity of your body, the structure that collagen provides, metabolism is a universal theme of your life. Everything good or bad, again, happens with metabolism. And this idea that you can actually look at 2 of the key metabolites. If you're a biologist, you know FED and NADH well. They're essential coenzymes that are needed for metabolic progression. But again, nature was kind enough. They made them really fluorescent and made them very fluorescent. They're very easy to image and very bright on our microscopes. Using any 2-photon microscope, even a confocal microscope can see these a little weaker. But you can excite these systems and be able to excite them. And then there was talk at breakfast today about the idea of the challenge of looking at mouse models. So as many of you know, with this kind of method, there's a little toy mouse, but we actually have to implant a window. And we've been trying to make windows that are better, windows that are not metal, windows that are plastic, 3D printed, so we can use them in MR machines. And more recent designs allow us to actually put things in there like temperature regulation, drug implantation. So the idea of having a little lab window, but you can implant this in the animal, in this case over the mammary ducts of the mouse, and the animal will actually survive just fine with this. It's actually the cancer that ultimately is its problem with health. And over the course of 3 to 4 months, you can watch this progress over the mammary. And similar work has been done in the brain and other models as well. And so by understanding this intrinsic contrast, so again, NADH can be seen label-free in the breast. You see the cells here, FAD as well. And then we can see the collagen I talked about as well. So in this case, you can compare FAD in cells with collagen. So you have the benefits of both worlds. And there's been a lot of work going back a long ways, back to the 90s, where you can use this technique of fluorescence lifetime I mentioned to track this, to look at lifetime. And a lot of papers have published in this. And basically the idea of this method is that you can combine your power of this intrinsic fluorescence by doing lifetime. And what lifetime can tell you is not only can it separate out the GFP or favorite marker from the NADH and FAD, but it can also tell you what's free or bound. And the real secret of NADH is it has a free and bound state that's absolutely vital to progression of disease. And it's been shown that this process called Warburg is a central aspect of all cancer. And the ability to see more free NADH is thought to be the shift towards a different pathway called glycolysis. If you're not a biology, a very simple analogy, again, one of my famous bad analogies, is if you're in a very crowded subway car and you're really hot and all your friends are hot, it's getting really hot in there, there's one weird guy in the corner wearing a sweatsuit, a hat, and gloves that's jogging up and down, and he's not sweating at all. That's cancer. Cancer can shift to a different metabolic pathway when conditions get bad. And that silly analogy tells us how bad cancer is. Its ability to, even in crowded conditions, outcompete us normal guys is why we're excited to be able to do cell-level resolution mapping metabolism. So we can essentially tell you what cells in this population are cancer, which ones aren't. And if that's all the talk was, there'd be some value. But a cancer biologist would say, you know, Kevin, I have other ways of telling cancer apart. There are known labels. There's other ways of telling cancer from normal. What's exciting is this hidden little graphic here, they'll go more depth, is that I have listed here 3 different labels. So you'll see a red, a green, and a gray. But you may be surprised that I'm starting to bring in a green here. I keep in this thing called CFMS. This is a probe for macrophages. I wanted to see what happened if I labeled macrophages and be able to see how it correlates to metabolism. My hope was not only could I tell apart cancer cells from normal cells in the context of metabolism, but could I tell apart macrophages? Because until now, usually in live cell imaging, we have a very hard time without specialized probes that are normally very error prone and normally ex vivo. We have a hard time telling things apart. And what we wanted to do was start looking at this sample and really understanding what happens with samples that are higher in FAD, which is one important coenzyme, and have a different change in NADH. And long and short of it, we want to look at this interplay, and then we want to map that to the known macrophage marker. And what we're able to show is by mapping this beautiful image, which is live in a mouse. So this is a live in mouse breathing. We're imaging it with multiphoton. We can not only see the collagen, these wispy white fibers, but what's really exciting is that we can see the lifetime of the NADH to red co-localize, means overlaps, with the green. And essentially, we found that the GFP cells have a unique lifetime from the others. So the idea that a cancer cell, a normal cell, and a macrophage are all unique. And the ability to metabolically map with that kind of resolution, not only in a single cell, but subcellular, where you can even see the cytoplasmic nuclei, is quite powerful. And being able to show what happens, so monocytes means macrophages, between these different cell types and the difference in FAD and NADH. And so now we have this ability where we can sort, and we can actually look at these changes in markers, particularly macrophages, in relation now, go back to the collagen model, and now we can label free so we can see which ones are cancer cells migrating along the fibers and which ones are macrophages that may be producing or regulating collagen. So that interplay. Because we had movies that were showing cancer cells pulling on collagen. We couldn't figure that out. It turned out they were macrophages pulling on the collagen, which makes more sense. So this idea of being able to discriminate in a live animal. And what's really exciting is that we also can use this to understand what might happen at the interplay of the collagen story and the metabolism story. I mentioned to you that more aligned collagen is a sign of cancer invasion metastasis. But what about Warburg? What about this idea that cancer cells are more glycolytic? It turns out that wherever we see the cancer cells at the intersect of more aligned fibers, we're also seeing that they have more glycolysis or more of this change in lifetime. So this ability to map those two is quite powerful. So concluding this part of it, this idea of tax, this idea of more aligned collagen as being a negatively prognostic factor, which means you're more likely to die with this. The idea that NADH, FAD, and harmonic can provide this beautiful endogenous fingerprint of what's happening in the stromal and tumor context. And that you might be able to use this method of lifetime to measure the free amount of NADH as a way of getting metabolic mapping. And we have since proven, because in other tissues as well, so we now have built over space and time to label free, tell you what metabolism is going on. And not only could we do this in cancer, we've even been able to show that a differentiating stem cell, when it started to become a cardiomyocyte, we have found one metabolic signature for that versus one that's not going to become a cardiomyocyte. So the idea that you can actually determine cell fate just by metabolism is what we're trying to do here, whether the cell fate be a tumor or a normal cell. And this idea that you can differentiate even further between NADH and FAD to further differentiate. So we have done all this work in a mouse model, and this idea that you look at collagen architecture, metabolism, and see the invasion is, again, another nice movie of a single cell crawling along a fiber to invade. Some of my favorite movies. It's very hard to capture, by the way. We'll listen to that a few times. A literal needle in a haystack. And this, again, is summarizing the earlier slide I showed, but this time showing with our data. So now the dynamics are the cells invading the matrix. The structure are this invasive, non-invasive 3D picture of collagen and cells. The collagen orientation label free. It turns out that's a big part of the stiffness. So getting the physics of the sample, the stiffness and the material properties, and then of course the lifetime with that. And in my last minute, I'm going to talk about what I often talk about, but don't have time today, is everything we do is image informatics based. And there's this whole amazing community developing software, and everything you do with an image requires software. And it is an essential part of imaging. And there's this wonderful world of open source imaging, which I'm part of, where we're trying to develop software that, yes, it's free, but more importantly, it's accessible. The idea that wherever you are, you can download this software. And we're trying to create a complete ecosystem where we develop software for each part of this. So when you're acquiring the data, we have Micromanager, that's free software that can acquire data. We developed a whole bunch of open friends, so OpenScan for laser scan microscopy. So if you're building a confocal microscope and want to do it without partnering with a major commercial vendor, it's not that we're anti-commercial. In fact, many companies work with us, is that we're trying to create open, transparent systems for trying new things. And we're fortunate to have Zeiss and many other manufacturers work with us. And open Polscope, where we can try to do polarimetry at the microscopy scale and actually look at changes in collagen, for example, with polarimetry. Open Spin, which is a way of doing light sheet. And then in the metadata side, bioformats, probably the most boring project our lab has ever done, but in the world of optical imaging, just in optical imaging, there's over 150 file formats. So we wrote a file converter that will take every file format out there, whether it's Zeiss or Nikon or Olympus, and we'll convert it to an open standard, usually based on TIFF. And then we've also worked with the open microscopy environment to develop a database of all these images so you can tag them and explore them. And then the part my lab mostly does is we develop ImageJ and then friends like Fiji, NIME, and SoProfiler to actually analyze your images, to basically look at cell changes, segment cells, and understand what they do. And if you're interested in anything, I'm pretty excited that over the last month, we've launched a new forum. We wanted a place for students like you or anybody at any level to ask a question. So if you're in this audience and you have an image analysis question, about 25 of us have created a forum for image analysis. And this is meant to be like the very famous confocal forum for confocal microscopy, but it's for image analysis. So it used to be that for every software package, you'd have to go to one of our sites, whether it was SoProfiler, an OME, or ImageJ, and ask your question. We've all gotten rid of our own individual listservs, and now there's one place you can ask. And it's like Facebook, very easy to post and ask questions. And then I will end there with all the many people that are in our group. We're always looking for students. We have an exchange program called the Bow Scholar Program, that if you're an undergrad here, we bring over Indian students fully funded to our university. Bose is a very famous Nobel Prize winner, Indian scholar. So if you're interested in that program, talk to me. I've had a lot of Indian grad students and postdocs, including 2 currently. We always love that people come over, and we are looking for people, so come talk to me if you're interested. And I'd like to thank our many students in our lab and collaborators, and all of you for listening to me on the last day at the first talk. So thank you very much.
The session is open for questions.
Kevin over here. Okay, great. Thanks very much for an interesting talk and beautiful images and lots of stimulating information you provided us. One quick question: when you talked about the tumor and basically tissue in general, I noticed in your discussion of cells and extracellular matrix, there wasn't much talk about fluids, about interstitial fluids, about lymphatics, about blood vessels, microvascular. You mentioned them briefly in the brain. Is that because you feel they're less important, or is it just something that's could you okay, maybe you can just comment on that. That was my question.
Absolutely. Purely a time constraint. That's a very active area of interest, is the intersection between the bloodstream is such an important part of the metastatic process. And with the collagen story, it's very linked. We think there is an interplay. We've been we're seeing the image blood vessels at the collagen. We're starting to see changes there. So for this biomarker talk in human pathology, we're often looking at biopsies and tissues where we don't see that. But in live imaging, we absolutely do. So with this kind of imaging and also with photoacoustic imaging, you can see dramatic changes in vasculature that correspond to a tumor. And really interesting, we see a change in vasculature corresponding with collagen. And we've also have seen, which I don't have in the talk, their cells do have a different glycolytic signature near a blood vessel versus not. And so that's also exciting. So yeah, absolutely, the whole exchange and of nutrients and the blood is a big part of cancer vision. So.
What about, do you look at lymphatics, or do you look at interstitial fluids, or at least acknowledging that there might be something there that you might need to look at?
So we started the area we've done the most is in the brain with looking at sleep. And we've seen a big change in the lymphatic system with that, and it's known that's very important. As far as breast cancer, we haven't done it yet, but we suspect there is. But definitely in the brain. So, good question.
If I'm allowed, I'll have one very quick other question. Sorry, you briefly mentioned that there are ways of measuring stiffness or mechanical properties of tissue with optics. Did you mean optical coherence elastography, or what? Could you expand on how you measure mechanical properties with?
There are several ways. So doing optical rheology, So there are ways with any method where we can actually apply stiffness. We can do optical rheology. We can use, there are known probes for stiffness. There's also harmonic itself, see the changes in collagen. So either doing optical rheology, harmonic, or some sort of probe is mostly what I was talking about. So, but again, a really exciting area because largely stiffness has been ignored in this kind of thing. And it's really thought that, it's amazing to me how you can make in a artificial gel, if you just put force, if you stretch a gel or stretch a tissue, or if you put in magnets and you just realign the fibers or cause them to be more dense or stiff, the cancer cells will start invading. With no road, no RAS, no biology there. It's a fake artificial model. It's just cells in a gel and they're more invasive, right? So that's, yeah. So the role of mechanics in cancer is still poorly understood. So. Anyone else? Go ahead, yeah.
Is it feasible to use the higher harmonics here as you are using SAG? So can you go for the higher overtones in the imaging process? Higher harmonics.
Yes, yes, yeah, it was a great question. I think I talked to people that work on ultrafast lasers, that like OPO lasers, this kind of thing, that there's this really, again, very poorly understood field of what happens when you do things like third harmonic or three photon. And particularly when you get to the higher order harmonics, similar to me omitting the blood vessel story, there's a really fundamental part of the collagen story I completely ignored for time. That's collagen cross-linking. So it's really thought that the cross-linking of the collagen is a big part of what Alex was referring to with the stiffness. Cross-linking is known to be so important, but for the most part in imaging, we're blind to it. Techniques that have been tried like CARS or polarization methods are largely blind. And we hope that methods like such as higher order harmonics might be sensitive to changes in cross-linking. So cross-linking is something we'd love to go after that. But yes, with a different laser type and optics, we could definitely do that. So.
And in one point, you mentioned that histograms can be biased by fiber density. So can you just elaborate on that? Histograms can be biased by fiber density.
The histograms can be biased by fiber density?
Yeah.
Not sure what you're referring to. You're referring to in the histogram of breast density, or?
Yeah, over there. That is the breast.
Yeah. Maybe what you're referring to is just a lot of context is the more and more dense fibers you have can really cause problems in reading the images. And so they have a problem with traditional methods that they really can get obscured images. They have a hard time diagnosing the patient. And with our method, we can get much higher accuracy. So we are able, I know what you're talking about. You were talking about how, yeah, normally a pathologist or a human can actually get wrong readings in collagen fibers. And I was describing as our method is less biased because it's an algorithm that is measuring the fibers, not a human. So there were two kinds of bias. There's one bias I had early on, was talking about the challenge of medical imaging of getting accurate results. But I think the question you're referring to was that when you have a human measuring this, there can be some bias because it's very hard to measure all those fibers by hand. And with automated software, we're more accurate. And we've been able to prove that by creating computational phantoms where we can artificially draw all phantoms in known angles, and we can see how a human compares to a computer program. And that's what I was referring to. So.
Thank you.
Any further questions? Thank you, sir, for this wonderful talk.
Thank you very much.
I now request Dr. Preetham Dev, Associate Professor, Department of Physics, Tezpur University, to present a memento of gratitude on our behalf to Dr. Elisuri. Thank you, sir. It's an honor to now introduce next speaker for the day, Dr. Shyam Prasad, Associate Professor, Department of Cell and Molecular Biology, School of Life Sciences, Manipal Academy of Higher Education. His field of expertise is epigenetics and cancer. He has an experience from the past 14 years with 39 publications and 1 patent. Dr. Sham secured the second rank in post-grad exam in MSc Biosciences from Mangalore University in 2004. He received the Best Poster Award at Mahe Scientific Meet held on 5th January 2008. He also received the Young Scientist Award 2011 by Indian Society of Human Genetics on the paper entitled Aberrant DNA Methylation Profile and Frequent Methylation of dog2b Gene in Carcinoma of Human Uterine Cervix at International Conference on Genomics, Genetic Diseases and Diagnostics and 36th Annual Conference of the Indian Society of Human Genetics held from Feb 14 to 16, 2011 at Mahe. Dr. Sham also received the International Travel Fellowship by DBT Government of India to attend 10th Asia Pacific Conference on Human Genetics held at Kuala Lumpur, Malaysia from 5th to 8th December 2012. He received the Certificate of Appreciation for Young Investigator Award at 10th APCHG held from December 5th to 8th, 2012 at Kuala Lumpur, Malaysia. He has been awarded with the Invention Award by Intellectual Ventures India, Bangalore for the invention entitled Increased DNA Methylation of Specific Genes and Decreased Expression of Corresponding Proteins as Novel Non-Invasive Diagnostic Biomarkers for Cervical Cancer. Dr. Sham has been a TMA Pi Gold Medalist, receiving the Research Award 2014 by Mahe. He has also been awarded with the Young Scientist Award WIFRA 2015 by Venus International Foundation, Chennai, Tamil Nadu. He has been a featured scientist of Mahe 2018 and also the editorial manager, Public Health Genomics Journal from 2015 to 2017. Today he'll be talking on confocal microscopy, its applications in life sciences. I welcome you on stage, sir.
Thank you. Good morning, one and all, and thank you for the nice introduction. And at this outset, I would like to thank Mohta sir and Nirmal for giving me an opportunity to come and share some of our experience with confocal microscope. So the title of my presentation is Confocal Microscopy from Fundamentals and its Application in Life Sciences. So what I'm going to do is for next 45 minutes to set what is confocal microscopy, what is the principle behind that, and where people are using. And second aspects will be some of our results or ongoing study where we are using this machine. So mostly I will be speaking majorly on the principles and how in life sciences we use this machine for getting cell biology related applications or information. So before I go into my presentation, I would like to introduce our school for the benefit of non-Mahe members. So I'm from School of Life Sciences, and we offer courses in BSc Biotechnology. Then we have courses in MSc Molecular Biology and Human Genetics, Medical Biotechnology, Bioinformatics, then MSc by Research, as well as PhD program. And we are going to add new course on MSc in System Biology from this year onwards. And we have about 7 to 8 departments, starting from the Aging Research Unit, Biophysics, Biotechnology, Cell and Molecular Biology, Plant Sciences, Radiation Biology and Toxicology, Bioinformatics, and Department of Public Health. And we take cutting-edge research in the area of genomics and human disease, epigenetics and cancers, genomics of human variation, and others. So coming to today's business, so it's about the microscopy and how we can use different types of microscopy in the field of life sciences. So you know that there are objects which we can detect by our naked eye, and there are some objects which we cannot detect by our naked eye. In such cases, we have to go for the concept of microscope. So if you see the microscopy, so you have a light microscopy and electron microscopy. Depending upon the size of the object which you are under investigating, so we can use different approaches. So what I'm going to do is talk about more on the confocal microscopy. So this is about the history of microscopy. We know that Antony van Leeuwenhoek, he built a simple microscope in the year 1674, and from there a lot of efforts and research has been taken with respect to the instrumentation of the microscope, with respect to the optics part, with respect to the how we can integrate computational part to the microscope. So we have different types of microscopes which have been generated from time to time. So you can see in so it was in 1625. So there is a first use of microscopy has been dated, and from there there is a advancement, and now we have most advanced microscopes where we can image, we can do live imaging and many other things. And if you see the microscopy, so we can have optical microscopy, electron microscopy, and scanning microscopy. So I'm not going to the details of this, but I'll be focusing more on how microscopes have evolved and where, how and where we use the confocal microscopy. So this is the simplest microscope, Antony van Leeuwenhoek has invented, and this is the microscope we have at the School of Life Sciences. We have a SP8 model from Leica Microsystems, and with the softwares for various image analysis. And he is the person, Marvin Minsky, in 1957, who brought the concept of or the principle of confocal imaging, and this is his block diagram. So I'm from a pure biology background. I know how to use this instrument. So if you ask me what happens in this block diagram, I won't be able to answer. So but what I do is I use this principle for addressing many of the questions related to cancer such as cervical, oral, and breast cancer. And if you see the imaging techniques, as the microscope build itself, we can classify it into upright microscopy, inverted microscopy, and stereo microscopy. And if you see these upright microscopy or inverted or microscopy or stereo microscopy, we have technique per se. If you see, it comes under optical microscopy, confocal microscopy, then transmission electron microscopy, scanning electron microscopy, and atomic force and scanning tunneling microscopies. And if you see how it happens or the images are formed, if you see the optical microscopy, light rays are used. Confocal microscopy, we use a coherent light source such as laser. Then we use electrons, and then we also use molecular mechanical probes. Okay, so we will talk more about this confocal microscopy, and I'll tell you why confocal microscopy is important in our subsequent slide. So what do you mean by a confocal microscopy? So how many of you have heard about confocal microscopy? Okay, so many of you know about the confocal microscopy. So how many of you know the principle of confocal microscopy? Okay, so few of them, you know the principle of microscopy. So here, what we do is in a regular microscopy, we use, such as fluorescent microscopy. So the entire specimen is flooded with the light source. So what happens is that it gives a lot of unwanted excitation as well as emissions, as a result of which you get a lot of background. So in contrast to this, in confocal microscopy, what we do is we use the principle of point illumination, and we use something called as pinhole. This is what we use, which is different from the regular fluorescent microscopy, the use of point illumination and use of pinholes. So basically, these pinholes are in conjugate to the focal point of the lens. That's why it is called as a confocal microscope. I'll tell you what exactly this conjugated to the focal point in the subsequent slide. So based on these two words, conjugate to the focal point, so this microscope got the name confocal. So how it is different from the other microscopy is based on the way the light sources are built and the way we have the pinholes, which prevents the incidence of an unwanted light, as well as the prevention of the unwanted light which is coming from the specimen. So that's how we get a very bright, crystal clear images. So if you see the principle, so we should have a element. So the principle of confocal microscopy, what we do is we eliminate out of focus light. Okay, so how to eliminate out of focus light? So we use a light source which is a point illumination. So wherein specific area of your image is being shined with the help of a laser, instead of shining the entire specimen. It is like something like this. If you imagine that this is the specimen, so you can see this projector. So it shines the entire area of the or the specimen which you want to image. If you talk from a confocal microscope, you can see this pointer, so it will do a point by point imaging. So that is the major difference between our confocal microscopy and regular microscopy. So herein we eliminate out of focus light, producing a very high resolution Z imaging, okay, or Z imaging. So we can do X, Y, T, and Z imaging in case of confocal microscopy. So in the confocal microscopy, first thing what we do is, so we prevent so incident light is particularly focused on a specimen. So with the help of a small aperture, which we call it as a pinhole. So there is a first pinhole, and followed by, so we use lot of fluorescence material, so it will get excited and it will start giving the light. So when you collect that, when these lights are being collected by the detector, we use again one more pinhole, so that out of focus light is prevented. So basically in confocal microscopy, like other imaging imaging applications, so when it comes to the practical aspects, you have to prepare the specimen, you have to stain with fluorescence material, or it should have a autofluorescence. Then you should do a mounting, then you should do a image acquisition, you should do a data analysis. So don't think that these steps are simple. So you have to take utmost precautions in each and every step of the confocal imaging, so that we get a better high resolution images. So I'll tell you why what are the precautions we have to take. So this is an image, okay, where you can see that this is captured with the wide field microscopy, whereas this is captured with the confocal microscopy. So can you tell what is the difference, audience? So what you see the difference here? So you have a better resolutions. So things you can see very clear as opposed to the wide field microscopy. So why you have this kind of problems in the wide field microscopy or regular fluorescent microscopy? Because you are illuminating the entire specimen with the help of a light source, so that you are collecting the information which you want as well as which you don't want. So you have a very hazy image, so which can be avoided by having a pinhole. So and you get a crystal clear images. So these are some of the major similarities and difference between the confocal microscopy and a regular fluorescent microscopy. So you have a wide field fluorescent microscope and you have a confocal microscope. In the machine itself, so what we have is here you have a fluorescence light source, then you have a camera, and you have filter cubes, fluorescence filter cubes to collect the information. Whereas in case of confocal microscope, it comprises of a laser light, then you have a pinhole, then you have a photomultiplier tube or PMT as a detector to collect the information. So here the principle is entire specimen is bathed with the light, whereas only a single point is illuminated with the light in case of a confocal microscopy. So as a result of which, in the wide field microscopy we have a low resolution, whereas in case of confocal microscopy we have a very high resolution, which leads to a blurred image in case of wide field microscopy, whereas a very high contrast image or reduced blur image in case of confocal microscopy. And these are the other aspects of the difference between the confocal and wide field microscopy, which we will see in the subsequent slides. So what are the advantages of confocal microscopy? You can have a more color possibilities. What do you mean by a more color possibilities? More parameters can be investigated. So if you have a microscope such as SP8, what we have, 8 lasers can be coupled in the machine, and 32 parameters simultaneously can be imaged with the help of an advanced microscope, which is little difficult with the help of the wide field or fluorescent microscopes. So you have a less crosstalk. So that is one more advantage of the confocal microscopy. Then you have optical sections of the object can be done without physical contact. Then three-dimensional reconstruction of the specimen can be formed. More importantly, you have a improved resolution. So let us see how these things can be done. So you have, as I told you earlier, we have to prepare the specimen, we have to stain, we have to mount, we have to do the image acquisition, we have to do the data analysis. So I'm going to tell you more about what are the precautions we have to take and how to do this experiment. Okay. So what are the, before I go into the details, so what are the applications where we can use it for localization and co-localization of the proteins. So for example, so if you see from a cell biology perspective, it is not the how much protein is present, in where it is present, or the compartment of the protein is important, and what stage it is present, whether it's a phosphorylated form or unphosphorylated form, whether it's a ubiquitinated form or it is a somolificated form or glycosylated form. Those forms are more important than merely having a the number of proteins or the amount of proteins. So a protein does its function by doing a compartmentalization. When I say a compartmentalization, it refers to whether it is localized to a mitochondria, whether it's localized to a chloroplast, or whether it's localized to a plasma membrane, without which these proteins are inefficient or they are non-functional. So these things can be identified by localization. What you mean by co-localization? Co-localization means, so these are the applications where you can use it for protein-protein interaction kind of studies. Okay, so a protein can be interacting with the other proteins can be investigated by co-localization and other applications. So I'll tell you what are those applications. Then you can use it for imaging of 2 or more fluorescent stains. Then it is possible to do a 3D visualization, then quantification of the protein expression, then acquisition of high quantities image. So this is the architecture of a regular microscope, where you have a sample, you have the objective, you have a dichroic mirror, you have a sample source, and you have a camera. So and you capture the image. So as opposed to this, you can see the confocal microscope. So what are the difference here? What you can see? So you can see a pinhole. Okay. So where you can see the pinhole, so you can see a pinhole here and you can see a pinhole here. So what is the purpose of using pinhole? To prevent the incidence of an out of focus light as well as or unwanted light, and prevention of a out of focus light, so that you get a very clear, crystal clear images. Okay. So what are the confocal imaging modes? So we have this is a triple labeled image. I will talk more about that in the subsequent slides. Then you have a time lapse imaging, means you can observe the accumulation and degradation, or accumulation and moving of certain protein in and out over a period of time. Then you can do a optical sectioning. So you can take a section from different areas, then you can merge them together so that you get a complete picture of that particular specimen of your interest. So these are the different types of imaging we do, and we can do it for fixed cells as well as live cell imaging. So one of the prerequisite, or it is important to have your specimen which is fluorescently labeled, or your cell should give an autofluorescence which can be captured with the help of a confocal microscope. So when it comes to a immunofluorescent staining, so what we do is we use an antibody, or we use some proteins which can give fluorescence. For example, you can clone your gene of interest in a GFP or RFP vector, and when the proteins when it is transfected into a cell line, so what happens is that these proteins, when it is getting expressed, so it gets tagged with the fluorescent proteins such as GFP or RFP or YFP or M-cherry. So it depends on how many molecules you are doing or how many proteins you are investigating. You can use different types of fluorescent molecules to be coupled, or you can use live staining, live imaging stains, such as, for example, if you want to image mitochondria, we can use mitotracker stain. If you want to use endoplasmic reticulum, you can use ER tracker stain. If you want to look into the lysosome, then you have a lysotracker. So all these trackers can be used. Okay. So sometimes what happens is that you don't want to use any transfection or with the help of proteins, and you want to capture the image. So in such cases, what we do is we do something called as immunofluorescent staining. Okay, wherein we use an antibody which is specific for a particular protein, and then you use a secondary antibody which is fluorescent labeled, and you capture them with the help of a fluorescent microscope. For example, you use here in this case we use confocal microscope. So what happens when you use this kind of immunofluorescent staining? So primary antibody binds to your antigen, and this antigen antibody a secondary antibody which is coupled to a fluorochrome. And so you can also do direct staining by primary antibody itself by labeling. Okay, so you can label the primary antibody itself, and you can introduce it into the cell. Okay, and then these kind of immunofluorescent staining are suitable for frozen non-fixed tissue and ethanol fixed tissue, or paraformaldehyde fixed tissue, or methanol or acetone fixed cells. I'll tell you I have two sections in the subsequent slide. So this is the basic staining techniques. So you have to prepare the cells, or you have to prepare your specimen, wherein you take you have to use cell culture work, where you grow the cells of your interest in a cell culture plate which is having a glass bottom Petri plate, or you you take a cover slip and you modify the coat the surface of the cover slip with some material, and you grow the cells on the cover slip. So you can use 6 well plate, 24 well plate, 96 well plate, depending upon your applications. Then what we generally do is we fix the cells using fixative. It can be paraformaldehyde, methanol, ethanol, or formalin, depends on what type of material which you are using. So accordingly, you have to tweak your experimental condition depending upon what fixative you are using. And the selection of the fixative is very important. I'll tell you in the subsequent slide. So what type of fixative needs to be used and when. And so so if you see the cell, so you have a cell wall, then you have cytoplasm, then you have a nucleus. So some proteins are localized to the plasma membrane, some proteins are present inside the your cytoplasm and in the cellular organelles, and some proteins are present inside the nucleus. So if you are imaging a protein which is present in the plasma membrane, so you don't have to permeabilize it. Without permeabilization, you can do the imaging. While if you want to image a protein or a molecule which is present deep inside the cell, such as nucleus, so we do something called as permeabilization. So when you do permeabilizations, these molecules are taken inside the cell and it goes to the respective, or it has an affinity towards the protein of your interest, and it goes and binds. And when you do your experiment, it starts giving the fluorescence. And then the third aspect of your this technique is immunofluorescent staining. So here what we do is we incubate with the cells with the blocking solution, and then we use a monoclonal antibody or polyclonal antibody specific for the protein of your interest. Then you use a secondary antibody, which is schematically shown here. So there is an antigen. So this antigen is recognized by a primary antibody. Then it is recognized by a secondary antibody, which is having tagged with the fluorochrome. You can use Psi3 dye, Psi5 dye, or Alexa kind of dyes. Okay, so these are the dyes which are commonly used. So the selection of the dye should depend on what type of, how many parameters you are going to use, and what type of excitation source, and what type of light collecting devices you have it in your machine. So what are the steps for successful confocal imaging? So you have to control the refraction of the light between your sample and the confocal pinhole. Then you have to choose an appropriate sample for the confocal microscopy. Then choose the correct objectives and immersion media. You cannot use every single immersion oil for imaging. Okay, so you have Leica, they have their own specifications. Carl Zeiss, they have their own specification. Olympus, they have their own immersion media. So you cannot use every immersion media for every type of samples. I'm going to tell you more about that in the subsequent slide. Then you have to choose a correct cover slip or sample dish. So the thickness of the slide what you are going to use, or the slide which you are going to use, matters a lot. Then you should, if you are going for a 3D imaging, so you should avoid compression and distortion of the samples. So otherwise it is going to, if you introduce an artificial force, then it is going to have the difference in the native architecture of the samples. Then you have to prepare the samples into an inverted light path or upright light path, depending upon what type of microscopes you have. Then you should have match the samples and its surrounding medium by the refractive index. So during this time, this immersion media plays an important role. Then more importantly, understand the spectral profile of your fluorochrome. When you are doing a multi-parameter imaging, this plays a very important role. So you have to select the dye in such a way that they have they don't crosstalk with each other, or they don't show an overlapping spectra. And that is what you have to take care of when you have to do a successful confocal imaging. So how to dye selection and sample preparation. So you have to determine the molecular association, transport, and metabolic fate of the probe. So you use a dye if it is impermeable to the cell, then you cannot use that dye. So when you select a dye, you have to select in such a way that it should transport inside the cell. It should be, you should know the metabolic fate. You use a dye, and if it is immediately, if it is quenched by the internal molecules what we have inside the cell, you cannot use such dye. Okay, then you should also know the molecular association. Molecular association means if you want to image a mitochondria, it should go and bind to the mitochondria only. It should not go and bind to the endoplasmic reticulum or Golgi body or other membrane bound organelles or organelles which is present inside the cells. So if it binds, then you will not be, the specificity and sensitivity of your experiment goes. So these things you have to be very careful. So the selection of the dye doesn't mean that you can randomly select any dye and you can use it for your experiment. Then, so water soluble dyes, yes, it is. If the dye is water soluble, so such dyes you should select. Then you have to see the membrane permeability, then receptor binding affinity and enzymatic conversion rate. So all these things I told you in the first point. Then, so these are some of the useful resources you can use to for your day-to-day research activity. So if you are working with fluorescent microscopy or confocal microscopy, so you have a probes for cytoskeleton proteins, organelles, then lipids and membranes, endocytosis, exocytosis, receptors and ion channels, then major divalent molecules such as secondary messengers such as calcium. Then you have magnesium, zinc ions, and other metal ions can be detected. pH can be detected. Then other molecules, salts, ions can be detected such as sodium, potassium, chlorine. Then probes for membrane potentials. All these things. So Life Technology, which is a company which is having a division called as molecular probes. So they have a nice catalog. So if you have a time, you please go through that catalog. So they have given vast number of dyes which can be useful, and they have given some principle how to use all the methodology, and everything is given in such a useful guide. So you should refer this guide. So coming to the first point, how to choose the correct dye. So these are some of the important points you should always remember when we talk about the selection of the dye. So you should use a dye which is of appropriate excitation wavelength. Then it should have a high quantum yield, then narrow emission spectra, minimum susceptibility to photopleaching, and then minimal disruption to the cellular processes, high specificity of the label, target abundance is a determinant of the probe selection, then concentration of the fluorochrome. So when you select a dye, so there may be, it should be selected based on your microscope. So what excitation source you have and what type of light collecting device you have. So accordingly you have to use a dye. Then it should have a very high quantum yield. Okay. Then it should have a narrow emission spectra, so that when this narrow emission spectrum comes into picture when you want to do a multiple simultaneous imaging of multiple samples. So you have a less crosstalk between the or overlap between the dyes. So better is the sensitivity and specificity of your imaging. Then it should be it should show minimum susceptibility to photobleaching. So you use a dye, and if you shine it with the laser or fluorescent light source, it should not get degraded very fast. Okay, it should not bleach very fast. So the dye which is having minimum photobleaching effect, you should consider for your experiment. Then when you use a dye, if it induces some kind of distortion to your internal cellular organelles, so such type of dyes it is not recommended for live imaging. So a dye should be selected in such a way that it should not inhibit, or it should not induce any mechanical or some kind of distortion to your cellular processes. It should not inhibit. Suppose you are using a fluorescent molecule, and if you inhibit the glycolysis or some pathways related to mitochondrial functioning, you cannot use it for a mitochondrial imaging, because the dye itself, although it is a very good dye for imaging, so it actually induces some kind of alteration in the signaling pathway, which will in turn reflect in the morphology of the imaging under investigation. Okay, then high specificity of the label. So if you are labeling, it should label what you are going to label. So if you see the confocal microscopy system, when it comes to the imaging, okay, so this is how it goes. So you have a sample, okay, then you have a cover slip, Then you have a immersion media. So this I'm talking about the 100x objective, or the oil immersion objective, which is 63x or 100x. Then you have a objective lens. So when you want to do a imaging, so this setup plays a very important role. So what you have a sample. Sample gives some kind of information about what type of immersion oil you have to use and what type of the object you have to use. Okay, then immersion oil will tell you, so based on the objective, sorry, it's a sample, so you have to use specific immersion oils. Okay, you cannot use some, we can use water, some we can use oils, some kind of oils. Okay, some specimens you can use DPX. Okay, I will tell you how to select the oil. Then you have to use a right objective for right imaging applications. Then the thickness of the cover slip also plays an important role when you want a very good imaging. So you cannot use a cover slip which is about 170 micron meter thickness. Then your image looks like regular fluorescent microscope. Okay, so idea of confocal microscopy is to get a very high resolution imaging. For that, so all these parameters plays very critical role. Okay. So coming to the cover slip thickness, generally, so objective lenses which we use for confocal imaging, they are designated, or they are having a thickness of 170 microns. Okay, so if you use a cover slip which is above this, or which is not close to 170 micron meter, so what happens? So it induces mismatch. So what are the mismatch? So mismatch related to getting a blurred image. So you will lose the information what you want to collect. So how this happens? It can introduce spherical and chromatic aberration. It can do spreading of focus light. So the idea of using a pinhole is to prevent the out of focus light. So, but if you don't use a right thickness cover slip, so what is going to happen is it is going to spread the light. Okay, then you will have a loss of resolution, the intensity decrease, distortion of the structures inside deeper layer of the samples, because you have to tweak the object, the adjustment in such a way that you are going to introduce a mechanical pressure on the sample. So all these things, so what happens is that it is going to give you a bad quality image. So sometimes, so to prevent this, some of the microscope companies, so what they have given is lenses that are equipped with the adjustable collar for correcting different thickness. Sometimes, so this thickness of the cover slip can be to some extent corrected by adjustable collar in your objectives. Okay. So what are the different types of cover slip I generally get? So if you see the cover of your or the packet of your cover slip, the box will be named with 0, 1, 1, 5 and 2. Okay, so this refers to the thickness of the cover slip. So the thickness are generally given in millimeter. So if it is 0, it is about 0.08 to 0.12 millimeter. It refers to 80 to 120 micron meter. Then we have 130 to 160 micron meter, which refers to this one, number 1 cover slip. Then you have 160 to 190 micron meter, then 190 to 230 micron meter. So these are the different types of cover slip which are available in the market. So we have to use the cover slip accordingly. So now coming to the fixation of the sample. So what type of fixatives we have to use? So when we say that fixative, the ideal fixative should penetrate quickly, then it should act rapidly, then it should preserve the cellular structure without any artifact. Okay, so these are the 3 important things you should remember. This is like very important when you want to do a imaging, whether you want to just detect a protein, whether you want to see where it is localized, or whether you want to see them in a native form or in a 3-dimensional architecture. So all these things will be determined by the fixative what we use. Okay, and if you ask me which is the right fixative, there is no protocol or fixative which is perfect. So you have to optimize every fixative or fixing methods for your experiment. So So the fixatives, if you know, they work by two principles. One is called as a coagulation fixative and the other one is called as a cross-linking fixative. So what you mean by a coagulation fixative? So which uses coagulation as a protein coagulation as a principle, and we have methanol, ethanol, and acetone. So these are the fixative which comes under coagulation fixative, and it is easy to apply and reproducibility is more. Then preserves the antigen recognition site very well and produces profound shrinkage. So if you want to go for a morphological based information, then methanol or ethanol or acetone based fixatives are not preferred. Okay, so in such cases we have to go for a cross linking fixative such as glutaraldehyde, formaldehyde or paraformaldehyde. So they preserve the cell structure very well. Then permeabilization steps are necessary. So if you see many protocols which uses methanol, acetone or ethanol, generally sometimes or many of the times they don't require any permeabilization step. Okay, so this fixative can do both the function of fixing as well as permeabilization. While if you see the cross-linking fixatives such as glutaraldehyde, formaldehyde or paraformaldehyde, which is mainly used for looking into the morphological aspect, so you require something called as permeabilization. Generally, we use specific concentration of triton X100 T 20 along with this one paraformaldehyde for based experiment. Sometimes people also use these what is that permeabilizing agents along with methanol, acetone, and ethanol based experiments as well, but not necessary. So coming to the mounting media. So when we discussed about the confocal microscopy, the imaging component. So we discussed about the cover slip. So now we are going to come to the immersion media. So you cannot use any immersion media for imaging, especially when you want to get a high resolution imaging. So if you see the mounting media, okay, such the common used mounting media includes water, then 100% PBS, then vector shield with the 50% PBS. So these are the Movial, which is most commonly used. You can even make this homemade this one, which we use for many of the applications. Then DPX we use, then glycerol we use. Then there are some commercially available. These ones are also available mounting media. So the mounting media has got refractive index, and when you want to use it for imaging, so it should closely match with the refractive index of your tissue or organ. So sometimes what we do is, without considering this, we use same mounting media for imaging, okay, every specimen. So but you should not do that. So even with the fluorescent microscopy or confocal microscopy, you should use what is that mounting media which is having a very close refractive index as your samples. For example, you have a spleen which has a refractive index of 1.4. Then you have liver, it's also around 1.4. Then you can see, so most of them are in around 1.4, 1.5 and 1.3. So you should select a mounting media which is having very close refractive index as your specimen under investigation. Then how to load the samples or the dye for the live imaging. So you have to label the specimen, so in such a way that the labeling should be very fast, it should be permeable, okay, it should not be photo bleached very easily. So what we generally think that increasing the labeling increasing the concentration of the probe may increase the good labeling. No, so you should identify the right labeling. What is that concentration? For example, if you are using a mitotracker dye, if it's a mitotracker red, we generally use from 5 nanomolar to 50 nanomolar. So if you use lesser than 5 nanomolar, so it doesn't stain the cell properly, and if you use more than 50 nanomolar, so it has got some kind of non-specific binding. Okay. At the same time, if you are using a lysotracker green, so what we do is we use around 75 nanomolar to 100 nanomolar. In that concentration we use. Both the dye stains the mitochondria, but they require different concentration. Okay, above the that particular concentration, so it has some drawbacks. So it leads to phototoxicity by formation of oxidate oxygen radicals, free radicals being generated. As a result of which there is a cytotoxicity. Then there will be a, if you have too much of the dye, then you will have a non-specific localization. Then it will also induce physiological or structural perturbation. So you should consider all these things when you select a dye for live imaging. Then what are the loading methods for live imaging? So you can use direct permeable molecule, you can use membrane permanent ester or permeant esters you can use peptide mediated uptake can be done, liposomes can be used, transient permeabilization can be done, osmotic permeabilizations can be done, electrophorations can be done, gene can be used, micro injection can be done, or whole cell patch recovery can be done. So if you this electrophoration means, so we generally go for transferring a plasmid into a cell, or we can use transfection, which is basically a liposome based method. So lipofectamine or any other molecules can be, your plasmids can be conjugated with those lipid molecule and it is taken inside the cell. Then you can use direct permeability. Direct permeability means you use certain dyes. For example, you have mito trackers, all these trackers. So from the media, it is taken inside the cell through the plasma membrane pores which are present in the plasma membrane. Sometimes what happens, you can use peptide mediated uptake. So these are the different modes which are available for loading the cells for live imaging. So now coming to how we do this imaging. So what are the devices which we use for the imaging? So there are different companies which use glass bottom or special plastics of very small thickness, which I mentioned in the previous slide. So that people use. So if you see these LabTech chambers, so this is a single chamber slide, this is a dual chamber slide, this is 4 chamber, this is a 8 chamber. So the advantage is that, so they all have a glass bottom of a thickness what I have told you earlier. Then you grow the cells over the surface, okay, and then you can do your experiment. For example, if you use this kind of chambers, so you can do imaging of this. Then if you have a test sample, you have to do the test sample in a separate vessel and then you have to do the imaging. But if you have this chambered glass slide, so the advantage what we get is you can do a control your test different concentration of the test simultaneously in a given slide itself. Okay, so basically it's a modified version of your cell culture vessel. So then you have a 6-well plate. So these are from different companies. Okay, so alternatively you can see here, so instead of, these are very expensive what is that materials. So instead of that, you can take this kind of 96-well plate, or sorry, 6-well plate. You take your cover slip and then you can put the cover slip, and over the cover slip you grow the cells. Okay, the purpose. And some people what they do is they take 6-well this kind of 35 mm dish. Then they take a very hot metal rod. They make pierce this one what is that plate, and then they stick the cover slip with the help of some kind of glue, and they sterilize it and they use. Okay, homemade stuff. So, and these are commercially available. So when this costs about 35 rupees or 40 rupees, this costs you around somewhere around close to 400 to 500 rupees. So, and if this is made up of plastic, the cost is around 300 to 400. But if it is made up of glass, then it goes up to 800 rupees and odd. So, for as a cost saving procedure, you can do this. So, how to prevent the bleaching of the sample? So, the bleaching of the photo bleaching of the sample, all the the imaging when we do, we use fluorescent molecules. When you expose it to light, they tend to get degraded or they get bleached. Okay, so we use something called as anti-fade. So when you do use anti-fade, so it can be for fixed samples as well as for the live samples. See, if you see the live specimen, you can, why we cannot use this fixed sample specimen for live imaging? Anybody can tell? So can you tell why these molecules or these chemicals cannot use for live imaging? Any thought? Because these are all toxic molecules. So when your purpose is live imaging, so you have to do the imaging in live conditions. So if you use these kind of chemicals, they are toxic and they can either do a morphological distortion or they can even kill your cells. Okay, or it can induce a cell death. So if you see these molecules, so these are all naturally occurring molecules inside our cell. For example, keratins or carotenoids, then which is present inside in the plants. Then you have ascorbates, imidazole, histidine, then reduced glutathione, uric acid. Then you can use a vitamins E analog. So these are all being tested and it is being used. Okay, these are all very well tested molecules. You understood, no? So these are not going to have any impact on the condition of your cell. While fixed imaging, what we do is we use something called as NPG, hydroquinone, then PPD and DAPCO. These are all being added in the fixative when you are doing a fixing. Then coming to the sources of the autofluorescence. See, when we say imaging with the help of this confocal microscopy, we actually shine it with the help of laser, and then the cell will start giving the fluorescence. You also know that from the previous talk and by reading, you know that our cell also contains autofluorescence substances. Okay, so what are the autofluorescent substances? So we have aromatic amino acid residues, then reduced pyridine nucleotides such as NADH, NADPH, then flavins, then zinc, then chitones, then chlorophyll. So all these molecules, they have a natural tendency to autofluorescence or give fluorescence when you shine it with a particular wavelength of light. So if you are working with the same wavelength of dye, so then you never know whether it is coming from your autofluorescence or it is coming from your test. So is it a biological variation or it is a artifact? You never know. So in order to identify the true biological what is that variations or the fluorescence, what you should know is what type of samples you are using and what are the possible autofluorescence that cell can have, and how what type of dye I should select or use when I am working with human samples or any animal samples or plant samples. So plant has got a lot of chlorophylls, which gives generally lot of background. Chlorophylls gets excited. Okay, when you use a particular wavelength of light. So we have to be very careful when you are going doing the imaging. So you should know all these properties. Okay. So what are the applications where you can use? You can use it for compartmentalization, then photo bleaching studies, fret, frap, then film. So these are all different types of experiment which people do. So next 5 to 6 minutes, I'm going to stop my presentation. So before we go into the details of what we are doing, a small portion of our research work I'm going to present. So this is one of the gene which is our primary focus, which is called as double C2-like domain beta, and it's a family of double C2 protein, and it has 3 isoforms, DOCK2A, DOCK2B, and DOCK2 gamma. Basically, this gene was identified during the brain cDNA screening, and it is involved in neurotransmitter release, exocytosis, and insulin secretion, glucose-dependent insulin secretion, and insulin-dependent glucose uptake, and then exocytosis. And more importantly, this gene requires calcium for its function. This gene has got 3 domains, a monk interacting domain, then 2 calcium domains, C2A and C2B domain. With the help of these domains, it does many of the functions which are involved in neurotransmitter, and it's related to a glucose and insulin secretion and related thing. And this gene is now considered as one of the potential molecule for diagnosis of diabetes. So in diabetic people have seen the downregulation of this gene, as a result of which there is no proper insulin and glucose based interactions. Okay, and we for the first time we showed that this gene is silenced in cervical cancer and oral cancer by a mechanism called as methylation. Okay, so we did a series of experiments. So as from a cell biology perspective, my interest lies in whether this gene acts as a tumor suppressor gene. If yes, so what are the hallmarks of cancer it targets? So for that, I have to develop some model system. So the model system what I have developed is retroviral and lentiviral based knockdown and overexpression models, wherein I have used SIHA and HeLa cells, which inherently doesn't express double this protein. So this is a good model for me to investigate the function of this gene, double C2 like domain beta. So what I did is I cloned my gene of interest in a retroviral vector. Then I have transferred this construct into a retroviral packaging system, Phoenix system, and then this Phoenix system produced the virus carrying this gene of interest. And then I took this, harvested this viruses and transferred it into my target cells. Means these are the cells. So now, once I do this, so I have to do some QC. So whether the transfer of my gene, whether it has worked, and whether it expresses the protein. So what I did is I did the RT-PCR and Western blot. So these genes I used as housekeeping, beta actin or actin, tubulin, anything you can use, where in both your vector transfected where you don't have a construct and your gene which is getting overexpressed, in both these housekeeping genes should get expressed. And so in the test means the construct with my gene, so it is getting expressed, but in the cells, parental cells, it doesn't get expressed. Then we confirmed with the Western blot. Similarly, we did the knockdown. Okay. So then we went on to investigate various hallmarks of cancer. You know that cancer cells proliferates, it grows faster, it migrates. There is a more angiogenesis, resistant to apoptosis, invasion is there, metastasis is there. So, and there is a aberrant signaling aspects which drives all these aspects. So we look both the aspects. Okay. So we showed that in the presence of this gene, there is a reduced tumor growth. Okay, which we quantitatively estimated. And then we did a xenograft study where we took the cells, control cells, and the doc2b expressing cells from both overexpression and knockdown model, and then we injected into a nude mice system. We grafted the tumor, and then we started measuring the tumor growth. So you can see here. Okay. So in the absence of the gene, there is a progressive and very big tumor as opposed to the cells which expresses the doc2b. So all these experiments showed me that it is a potent inhibitor of growth, tumor growth, proliferation, migration, invasion. So we did a lot of experiments, and since this is not the right audience for that, so I'm not going to share the details, but I'm going to tell where I'm using the confocal microscopy. Okay. So as I said in the beginning, it is not how much protein is present, where it present matters. So what we started doing is we started looking into where this protein is localized. So in the literature, it is known that in the other type of cells, such as in the brain and in the liver cells, and the pancreatic cells, what happens is that the gene is translocated to the plasma membrane in response to a stimulus such as calcium, or in response to glucose or insulin. This gene is transferred to the plasma membrane, but we don't know what happens in the cancer. So what we did is we did fixed imaging as well as live imaging, and what we showed is that by default, this gene in cancer, it goes to the plasma membrane. So you can see here. Okay, this is a confocal imaging what we have done. So these are the plasma membrane. This is a nuclear stain which we used as a DAPI, and this is a merged image. Okay, but in case of doc2b deficient cells, okay, so you don't see in the plasma membrane. Basically, this is a immunofluorescent staining. So in contrast to this, so we also have cloned it into a reporter vector, and you can also see the similar or the same phenomenon where it is getting translocated to the plasma membrane as opposed to the the doc2b deficient cells. So it indicates that in cancer, so this gene is predominantly localized in the plasma membrane. And then when we looked into this gene carefully, so what it shows is that it does inhibit migration and invasion of the cancer cells to the liver. So what we did is that, so when we looked into the gene expression microarray data of our cells, so we identified that epithelial to mesenchymal signaling and Wnt signaling, they are greatly impaired or downregulated in the presence of this gene. So when this gene is there, Wnt and EMT signaling goes up, whereas in the absence of this gene, Wnt and EMT signaling goes down. So what we have looked into is some of the major molecules. Actually, we have biochemically, with the help of Western blot and immunoprecipitation, we have characterized the entire pathway. But with the help of microscopy, so we have started this work. And one of the molecule is CTNNB1, which is nothing but beta-catenin, which is upregulated in many of the cancers. and which drives the growth, proliferation, which regulates many of these cancer hallmarks. So what we showed is that this is the in the presence of DOCK2B, all the beta catenin is actually localized to the plasma membrane, while in the absence of DOCK2B, they are localized to the nucleus. So we are currently working more on understanding this. And second aspect, we are looking this gene is from the mitochondrial perspective. So this gene is actually involved in biogenesis of various membrane-bound organelles. One of the membrane-bound organelle is the is the mitochondria. We did the confocal imaging for the mitochondria. So you can see here that this is the DOCK2B negative panel, and this is the DOCK2B positive panel in the overexpression system. This is the knockdown panel. So here you can see that all these green color thing is the nothing but the mitochondria. So a very well developed mitochondria you can see, as opposed to this, when you knock off overexpress this gene, so there is a change in the mitochondrial morphology, and there is a compromised function so in the mitochondria. So by doing this in one system, it may not be sufficient if you want to say that it's a universal phenomenon. So we also did a knockdown in another cell line, so where we showed that in the absence of this gene, there is a well developed mitochondria similar to this. So this is an analogous system. While you do the knockdown of when you don't have a knockdown, so you can see very fragmented or round shaped mitochondria. I won't say that fragmented mitochondria. There is a round mitochondria. So now we are doing some kind of FRET and fluorescence FRAP kind of experiment to investigate this further. And we can also use it for even looking into the mitochondrial DNA content. So what happens is that mitochondrial DNA content is reflect. It is also responsible for plays an important role as a retrograde or anterograde signaling molecule, which controls many of these nuclear encoded signaling pathways. So we wanted to see whether is there any reduction in the mitochondrial content. We did a real-time PCR based experiment, and where we show that in the presence of this gene, there is a tremendous decrease in the mitochondrial content. And then what we did is we also showed with the help of microscopy. So all these green color dots, so this big dot is nothing but the nuclear DNA. All these tiny dots are mitochondrial DNA, or the DNA which is outside the nucleus. Now, how to tell that this belongs to mitochondria? So what we did is we stained with the mitotracker dye, and then we did the overlap. So if this molecule is present inside the mitochondria, so what you can see is that a yellow color dot. So you know that red and green color, if you mix them together, what color you are going to get? You are going to get a yellow color. So that says that they are in a very close proximity, or they are physically interacting with each other. So we did that. And then this is from the published literature. So for the lack of time, I'm not going to the details of that. So you can do a time-lapse imaging also. So this is one of our own paper published by Dr. Joshi on it's called as NETs, or neutrophil extracellular traps. So here also we did some imaging, time-lapse imaging we did. And what we showed is that in a diabetic condition, there is a compromised nets or neutrophil extracellular traps happen. So I'm not going to the details of this. And so the previous part of the work is published in Journal of Biological Chemistry, and this he has published in scientific reports. And so to conclude, so we have the confocal microscopy is a powerful tool which every cell biologist and we use in understanding the localization, co-localization, and interaction kind of studies. And with this, I am going to stop my presentation. And at this outset, I would like to thank the organizers as well as our director, Dr. K. Satyamurthy, for setting up such an excellent facility, and our clinical collaborators for oral cancer and cervical cancer, and they provide the follow-up samples, Dr. Shorja Banerjee and Krishna Sharan. And then these are the present members and members who have already completed. So the major work is done by Samatha, and then we have Sanjeevan, he is a NGS and microarray specialist, and then Sandeep, he takes care of our bioinformatics facility, and these are our PhD scholars, and these are our newly joined team members. Thank you.
The session is now open for questions. Any questions from the audience? Okay, thank you, sir. Thank you for such an informative and wonderful talk. I would now request Dr. Kevin Elisuri, Director, Lab of Optical and Computational Instrumentation, University of Wisconsin, Madison Graduate School, to kindly present a memento of gratitude to Dr. Sham on the behalf of Manipal OSA Student Chapter. Thank you, sir. We now have a break for the next 15 minutes. You may proceed towards the backside of the building for tea and coffee. Some important announcement for all the delegates. All the poster presenters are required to take off their posters from the board during this break. Also, the participants of the activity hour from the first day may collect their charts from the registration room and display them on the poster boards upstairs. I request all the delegates to be here for a group photograph. Also, the volunteers should also be present here. The interested delegates for visiting the life science facilities can contact any of the volunteers present. Thank you.